The stems of woody dicots and gymnosperms grow in thickness. This is called secondary growth. In the stems of perennial plants, the central part of the procambial ring is transformed into a secondary meristematic tissue, the cambium. The secondary structure of the stem is clearly visible on a cross-section of a linden branch. The conducting tissues, xylem and phloem, form strands called vascular bundles. The inner part of a vascular bundle containing xylem is termed the woody part, and the part that includes phloem is the phloem, bast part. Those vascular bundles that adjoin strands of mechanical tissue, sclerenchyma, are called vascular fibrous bundles. They are clearly visible in a longitudinally sectioned stem of gradual touch-me-not. The bast consists of phloem sieve tubes through which the descending flow of dissolved organic products of biosynthesis passes, while bast fibers lie between the sieve tubes. The bark adjoins the bast on the outside. The cambium, which forms a ring, separates the wood from the bast. Because of its expansion, the stem increases in thickness, secondary growth of the stem occurs. Cambium cells contain numerous vacuoles and have thin cellulose walls with a small amount of pectin substances. As a result of cambial cell division, cells of the secondary xylem are deposited toward the pith of the stem, and cells of the secondary phloem, bast, toward the perimeter. The wood and the bast together constitute the system of secondary conducting tissues. In winter, the cambium is dormant, while in spring, even before the leaves unfold, sugar solutions are delivered from the root to the stem and its branches into the swelling buds, and the cambium begins to grow. Moreover, it deposits considerably more wood than bast each year, so the volume of wood increases rapidly. The wood is divided into concentric layers, each layer representing its growth increment over 1 year. Such annual rings are clearly visible on a cross-section of the stem, because wood is laid down unevenly in different seasons. The wood formed in spring and early summer is less dense, contains many vessels, and therefore is lighter in color. The wood laid down in late summer and autumn contains more mechanical and storage tissue and is darker and denser. The width of the annual rings varies from year to year. This depends on the amount of precipitation, temperature, illumination, length of the growing season, and ecological conditions. Under favorable conditions, wide rings form, under unfavorable ones, narrow ones. From the annual rings of many trees, one can reconstruct the climatic conditions throughout the entire lifespan. These questions are the subject of the science of dendrochronology. On a cross-section of a linden branch, the stem is seen to be covered externally by dead bark cells, behind which lie a brownish layer of cork impregnated with the fatty substance suberin. Beneath the cork lies the cork cambium or phloem, followed by the living cells of the protective tissue, the pheloderm, which contain chlorophyll. Next comes the living tissue with thickened walls that helps to reinforce the stem, collenchyma, and tissue composed of living thin-walled cells, parenchyma. The cork cambium is formed from the parenchyma, and from it, in turn, the cork is produced, which protects the plant from adverse external influences. Then comes the bast part of the bark, which in linden consists of alternating bast fibers, sieve tubes, and bast parenchyma. All of these are produced by the cambium, which adjoins the bast as a narrow strip. Next follows the wood, divided into concentric layers. At the center of the stem lies the pith. It is composed of large air-containing cells which cluster around smaller ones that reinforce the stem. The medullary rays vary in length and are made up of cells of approximately uniform size. The younger, light-colored conducting part of the wood forms the sapwood, while the older, darker part is the heartwood, which consists of dead cells impregnated with various substances that color and aromatize the tissues. The thickness of the sapwood differs among tree species. Thick sapwood is found in ash, birch, and maple. Thin sapwood in white acacia and yew. And sapwood not clearly separated from the heartwood in fir, poplar, and willow. The heartwood may be colored yellow, red, dark violet, or black. A distinction is made between hardwood possessed by broad-leaved trees, in which vessels, tracheids, mechanical fibers, and parenchyma cells are present, and the softwood of coniferous trees, in which there are no vessels, the cells are tracheidal, together with parenchyma cells associated with resin ducts. The term hard and soft reflect the structure of the wood rather than its density. Externally, wood is distinguished by its color, grain, cross-sectional texture, and figure. The grain depends on the arrangement and shape of the vessels, tracheids, fibers, and parenchyma cells. For example, when they are arranged parallel to the axis of the trunk, the wood is called straight-grained, when at an angle of the axis, spiral-grained, and when wound helically, spiral-grained, in which case the trunk without bark would appear twisted. Wood may also be interlocked grained if the direction of the spirals across the radius of the lock reverses at approximately regular intervals. Figure refers to the pattern on a longitudinal cut of the trunk, where the annual rings appear at various stripes. It emphasizes the decorative quality of the wood and depends on its grain and texture, as well as on the direction in which the cut is made.